Hi everyone, welcome to chapter 11. This one is entitled Muscular Tissue. In chapter 11, we're going to go ahead and explore skeletal muscle with a little bit more detail. We have at this point become familiar with the majority of the muscle names as well as their locations. And along the way, we also discovered the fact that within the human body, you have three types of muscles. We have our cardiac muscle, which we find solely within the heart. We have smooth muscle, which is mostly in our viscera or internal organs. And then, of course, we have our skeletal muscle, which is associated with the skeleton system and allows us for movement of our appendages as well as facial expressions, just to name a few. Now, it turns out that skeletal muscle has many unique features, so that is what chapter 11 is going to be dedicated to. We're going to take a look at what makes up the different anatomical structures within the skeletal muscle itself, and then we're also going to need to explore exactly how the nervous system and the skeletal muscle have to work together to get the muscles to ultimately contract as well as to relax. So before we get into the nitty-gritty details, let's go ahead and review some of the topics that we should already know for skeletal muscle. First off, we know that it's under voluntary control, and that means that on a conscious level, you're able to go ahead and control when the muscles will contract and relax. So that means, for instance, if we all take a second and look down to our hands, we can go ahead and make our fingers scrunch together into a fist and then relax it. Or we can consciously go ahead and kick our feet out, extend it, and then bring it back to our body. We also know that skeletal muscle is a striated muscle, and that means that if we're looking at it under the microscope, we're going to see that within the muscle fibers, we have these alternating light and dark bands that are parallel and very organized. Now, it turns out that these striations, these bands, are actually going to be our contractile units, meaning that we have to get these bands to work together so that on a larger scale we can see the muscle contracting and eventually relaxing. So let's go ahead and take a look at what's actually happening inside that skeletal muscle. Now, I do want to point something out, which is as we're going to go along, you're going to see different terminology when we're talking about the muscle. And that's because you want to keep in mind that a muscle is an organ, and it's composed of different cells. So the muscle cell is also referred to as muscle fiber or myofiber. And in fact, anytime you see the prefix myo, that means that we're looking at muscles. And don't worry, I'll break it down for you from large organ all the way down to the individual cells. This is an illustration that should somewhat look familiar to it. You actually came across it when we were looking at some histology, when we talked about the fact that if you look at skeletal muscle under the microscope, you're going to see individual fibers that are running together. Here's one, here's the second one, here's the third one. These are muscle fibers. So those are your little muscle cells. And within the fibers, you can see the alternating dark and light bands. These are going to be our striations. And part of what we'll explore in part one is exactly what makes up the dark lines versus the light lines. So what exactly is it made out of and how do they interact with each other? Here is another look at the skeletal muscle fiber. So now we're looking at our individual muscle cell, and as you can see right here, separated. Within the muscle fiber itself, what we see happening is that there are lots of bundles as well as smaller entities that we need to look at. Now, I understand that looking at this picture might look a little overwhelming, but don't worry, we're going to get to meet all the individual characteristics. And then what we'll do at the end is we're going to put them all together so that we can look at this picture again and kind of see how all of them work together. But for now, I just want to highlight the fact that your little muscle fiber is going to go ahead and be composed of these little myofibrils, these little bundles. And within them, you're going to notice that they went ahead and they did the color code. You have the little red line and the little purple line, or the thicker purple line. Those are going to be your myofilaments. Now, the myofilaments are what create the striations. So when you look at the muscle under the microscope and you see those little lines alternating, Those little lines are called myofilaments. And we're going to have a thick one indicated by the purple coloration and a thin one indicated by the red coloration. So, excuse me, let's explore them a little bit, and then we're going to come back to this picture and see how they all quote-unquote bundle together. All right, so let's start with the thick one. The thick filament that you see sticking out of your little bundles is going to be composed of a protein called myosin. All right, and it turns out that your body has different versions of myosin, but the myosin that we're interested in is going to be the one that's going to look like a golf club. It's going to have a tail section, and then it's going to have a head section that makes it collectively look like a little golf club. And the thick filament will have two chains intertwined with each other, and the head section is going to be pointed outwards, sticking out, and then the tail section is going to be making up the bulk of the overall formation. Now, as you can see, I have a little illustration for you here at the bottom. This is your thick filament, and for now, just keep in mind that it's going to look like a little golf club with the tail and the head section sticking out, and it's composed of myosin. Here we see it working together. Now, what's going to end up happening is that the individual myosin molecule will stack on top of each other, and as they start stacking, what we see happening is that the tails will flatten and the heads will keep protruding outwards. So collectively they will form the thick filament. So here on B you can see what your thick filament looks like, and as you can see you have all these globular heads that are sticking out. Now, the reason we care so much about this is because it turns out that when we look at the striations on the muscle, and we're seeing the perpendicular lines going up and down, very organized, what we're looking at is a thick filament followed by a thin, followed by another thick, followed by a thin. So it's very organized in the pattern. In fact, if you come over to the right-hand side of the illustration, if we take a look at part D right over here, let's take a look at the left side right here. You're going to notice that if we were to illustrate the striations that we see under the microscope, the thick one would be here, right here in purple. And then in between two thick ones, you have the smaller red structure that comes into play. You see it right here? Now, this red structure is going to be our thin filament. So if the thick filament looks like a golf club and it's made out of myosin, what exactly is the thin filament about? So let's click over to the next slide and let's take a look at that one next. All right, now our thin filament is going to be composed of actin. Once again, we're going to see that there's some intertwined strands, so there's two of them that are going to intertwine. And I'm going to ask you to go ahead and take a look at the illustration that's towards the bottom of the slide. And the actin is represented by the red balls. Okay, so here's where I have to tell you that when you discuss actin, especially when you take a look at it from the thin filaments, you have to be able to describe the difference between what we call globular actin with a G and fibrous actin with an F. Globular actin refers to the individual red circles. So the individual, think of them like little beads, the individual circles that you see on the illustration, the individual red balls, they are individually referred to as a globular actin subunit. Globular just means that it's circular. And when we string them together, we almost make like a pearl necklace. That strand, that necklace, is then going to be called fibrous actin. So the entire strand of the globular actins will go ahead and make up fibrous actin. So let me see if I can erase this real quick for you. There we go. So the entire strand right here, this is my fibrous actin. But then notice how they went ahead and they identified an individual red bead as your G actin. So your thin filaments are made out of actin. Now here's where it's going to get a little interesting. It turns out, and it's not really visible on the illustration that you have here, so I'm going to make my best drawing of my G actin, and you guys know how good I am at drawing, so bear with me. But it turns out that when you look at the individual subunits of the actin, they have a little active site. They have a little dip. And wouldn't you know it, the head of the thick filament, so the head of the little golf club molecule that we were just looking at, well, it just happens to fit perfectly within the active site of the actin. So this is me drawing my golf club of the thick filament, and that's the little golf club head, and it fits right into the active site of the actin molecule. Now, when your muscle contracts, when your skeletal muscle contracts, we want the thick filament and the thin filament to physically interact with each other. So we want the golf club head of the thick filament to interact with the active site on the thin filament. And when they link together, that is then what we see as a contraction. And we're going to go into this in great detail as we proceed through the chapter. But I just wanted to kind of point out what the importance was of these active sites. So right here in your PowerPoint, it says the active sites will allow for the head of the myosin molecule to bind. And remember, your myosin molecule is courtesy of your thick filament. Now, our muscles aren't always contracting. So there has to be a way for us to kind of regulate when the head of the myosin comes into contact with the active site and when it doesn't. So in order to do that, we're going to use tropomyosin and troponin. Tropomyosin-troponin complex is going to allow us to block the active sites on the actin so that the head of the myosin cannot make contact, aka we can't contract the muscle when we don't need to have it. So on your illustration, and what I'm going to do is I'm going to erase my little markings so that we can go back to our original picture. On our illustration, what you're going to see is that you have a elongated protein called tropomyosin. And here it is illustrated in this white formation. And what tropomyosin is going to do is it's going to block several active sites of the G-actin. So it covers the active site so that the head of the tropomyosin cannot physically make contact with it. In order to control the tropomyosin, we're going to need to use troponin. And troponin, illustrated right here in the yellow formation, think of it like a little doorknob. It is going to be able to control the movement of the tropomyosin on top of the active site. And when we need the active site to be exposed, the troponin is going to be able to move over the tropomyosin so that it can expose the active sites. So tropomyosin is the elongated protein that covers the active sites on your actin, and troponin sits on top of the tropomyosin almost like a little doorknob, a little door handle, and when properly stimulated, it will cause the tropomyosin to glide out of the way, exposing the active sites on the actin, and therefore allowing the head of the myosin to bind to it. This will ultimately lead to the muscle contracting. Now, what exactly is going to activate the troponin? Well, it turns out that one of the things that's going to activate it is going to be calcium. Whenever calcium is poured into the system, we see that the troponin will be able to notice it. It will be able to interact with the calcium ions, and it will activate itself so that it will move the tropomyosin out of the active sites, allowing for the interaction between the thin and the thick filament. Here is just another look at our thin filament, so we can see our actin molecules. We can see our fibrous actin because it's in a fibrous elongated string, and we can see our tropomyosin molecule covering several of the active sites on our actin, and then we have our troponin molecule that allows the tropomyosin to move on and off, exposing or protecting the active sites of the actin. We also have several other proteins that will keep the system regulated, meaning it will continuously monitor the distance between the thick and the thin filament and make sure that they are properly stacked together and are able to interact with each other when needed, meaning when the muscle needs to contract. One of those filaments that we rely on is classified as an elastic filament, and probably the most important one is titin. Titin will run along the core of the thin filament and anchor itself onto the edges, what we call the Z discs, and this will allow you to stabilize and position the thick filament. The reason this is so important is not only is it allowing the thick filament to stay in its proper location, it will also prevent the individual muscle cell to overstretch, and of course when it does stretch, to prevent any recoil that's going to occur when it needs to relax and go back to its at rest position. I'll show you how these pieces fit together in the next picture. All right, so here we go. So here we're looking at, once again, our thin and our thick filament stacked together. So these are the striations that we're seeing. Here's the thick filament in purple. Remember, that one's made out of myosin, and you see the little golf club heads sticking out. And then here is your thin filament made out of actin, and we talked about the fact that it has little active sites. So the heads of the golf clubs are trying to make a physical connection between the thin and the thick filament. And allowing the alterations to occur between thick and thin, if you come over here, you're going to see the elastic filament in green. And that, of course, is going to be stabilizing the thick filament in its proper place. Notice how the thick and the thin filaments are very uniformed in their organization. So they're alternating their pattern. And then the other thing you want to notice is that they're anchored onto this little wall called a Z disc. And what we see happening, and I'll show you this on additional pictures as we go along, is that from one Z disc to the next is what we're going to be calling a sarcomere, or a contractile unit of a muscle. So this is the smallest part of the muscle that you could get to contract from one Z disc to the next. That's what we're going to call a sarcomere. I know I'm throwing a lot of different names at you. We're just going to take it easy. We're just going to move along, and at the end of this part one, I'm going to have this beautiful chart for you that's going to show you how we went from a big muscle organ all the way down to all the small little entities that we've been talking about. So consider these few slides almost like a little introduction to see all these little pieces, and then we're going to start putting them all together, okay? So bear with me. So far, if you're comfortable with identifying a thick filament being made of myosin, looking like a golf club and having a little head section that sticks out, your thin filament is going to be made out of actin and has an active site that's protected by the tropomyosin and troponin complex. And our goal is to get them to interact, the thick and the thin filaments, when we want the muscle to contract, and to not interact when we want the muscle to relax. Then we see that when we stack them together, they're going to be coordinated from one Z disc to the next. And a Z disc, think of it almost like a little wall, a little perimeter. It's going to allow you to separate the contractile units of the muscle. All right, a small muscle will have a few contractile units. A very bulky muscle like our gastrocnemius will have thousands of these contractile units from one Z disc to the next. All right, so the contractile proteins, what's going to allow the muscle to actually contract is going to be the interaction of myosin, thick filament, and actin, thin filament. And then we're going to have regulatory proteins. These are going to be your tropomyosin and troponin. Remember, this is going to be the elongated proteins that cover up the active sites on the actin. And then the troponin is the little doorknob that sits there waiting for calcium to arrive so it can move the tropomyosin out of the way and therefore allow a physical contact to occur between the thick and the thin filament. So calcium is very important at this point because it will be one of the requirements to get our thick and our thin filaments to interact with each other. Let's take a pause and let's take a look at a clinical application. So when we're talking about the thick and the thin filaments, you want to keep in mind that we have just completely zoomed into the skeletal muscle, and we're taking a look at the contractile unit, meaning the smallest part of the muscle that needs to contract. And what we see happening is that when the thin and the thick filaments interact with each other, you get an amplifying result. So think about this, right? So let's assume that you just have one sarcomere, one contractile unit. So one little piece that goes from one Z disc to the next. If we get the thin and the thick filaments to interact, you'll almost get like a little twitch. But a twitch doesn't really do you a lot of good, right? You want to have the strong contraction. So what we see happening is that the muscle will have hundreds, if not thousands, of these contractile units that when stimulated will cohesively contract. And what we see happening is that the twitches then build up on top of each other and give us that smooth contraction and relaxation of our skeletal muscle. And this all starts from the internal workings of the skeletal muscle, and then of course it has to radiate out so it can interact with the sarcolemma or the outer workings of the muscle and get the actual bulk of the muscle organ on the outside to respond, meaning to contract as well. So there's this disease, or this protein, excuse me, that's called dystrophin. And what dystrophin does is that it basically links actin in the outermost myofilaments to the membrane proteins that link to endomyosin. Now, we haven't really discussed endomyosin just yet, but it is going to be a protective layer that wraps itself around the muscle. Picture coming soon, I promise you. And what we see happening is that the dystrophin protein is going to allow us to connect the thin filament that's on the inside of the muscle to the endomyosin covering that's on the outside of the muscle. So when the inner part contracts, the dystrophin is able to communicate that with the endomyosin so that the endomyosin will get the muscle as a whole to contract as well. Now, if there is a genetic defect with dystrophin, that means that you then lose that inner communication, which means that even though the actin and the myosin, so the thin and the thick filaments, are interacting and contracting, there is no connector protein that allows for the contraction to be displayed on the outside of the muscle. So hence, to the naked eye, the muscle will lay dormant even though its internal workings are actively trying to contract. Here is a look at what I'm referring to. So take a look at this. Let's start at the bottom of this figure. We have our thick filament, we have our thin filament, and as you can see right here in red, this is the dystrophin. So what the dystrophin is going to do is it's going to allow us to connect what's happening within the thin and the thick filament, which is happening on the inside of the cell. By the way, let's just take a quick moment. Notice how the membrane of the muscle cell is not called plasma membrane or cell membrane. Nope, we're going to get a special name, sarcolemma. So when you hear me say sarcolemma, that is just referencing to the plasma membrane, but a plasma membrane of a muscle cell, okay? So sarcolemma is just a cell membrane, but we give it a different name because it's exclusively for the muscle cells. So back to the dystrophin. It's going to connect the thin filament's movement with the sarcolemma, which then also radiates it out over to the endomyosin. And remember I said the endomyosin is going to be this fibrous layer that's going to be protecting or covering the outside of the muscle that we're looking at. So we're able to take the contractions from the inside to the outside. Hence, we can see the muscle physically contracting and eventually relaxing. If something happens with the dystrophin, then you lose the connection point, which means that even though the thin and the thick filaments might be contracting or might be interacting with each other, you can't communicate that with the outside, which means that the muscle will not be able to fully contract itself on a large scale because of the loss of that linker protein. Another way to think about the striations, and this really comes more into play when you're looking at it under an electron microscope is the fact that the layering of the thin and the thick filaments will create different areas. And what we can do is we can go ahead and label them things like an A band, an I band, an H band, an M band, a Z disc, and these will all reference different locational terms when you're looking at it through an electron microscope. Now for us, when we discuss it this way, the most important ones are going to be our A band, and let me just kind of like highlight this for you so we can take a look at it. It's going to be our A band, our I band, and our Z disc. Those are the ones we're going to be interested in. The A band, A stands for dark, is going to be the region where you have your thick filaments that are overlapping with your thin filaments. So by stacking the thick filaments over the thin filaments, it's going to obviously create a thicker area, which is then labeled as the A band, the dark band. The I band is where you're going to have your thin filaments by themselves. So it stands for, I is going to stand for isotropic, it stands for your light band, and basically it's light because there is a thinner formula or a thinner format in there, so the light will obviously reflect differently, allowing you to have more of a shaded area, creating a light band. Now, the combination of the dark and the light, the A and the I, will then be seen within the Z discs. Now remember, the Z disc is basically just the wall that will go from one Z disc to the next, will be our contractile unit, will be our sarcomere. And I know for those of our visual inclined, we're doing our best. Don't worry, a picture is coming up, I promise you. All right, so here's just a reminder of what we've been talking about so far. Remember, we have our thick and our thin filaments. And then we have our Z discs. Now, notice how on our sarcomere, right, from one Z disc to the next, that's the sarcomere. That's the contractile unit of the muscle. Notice how you'll have areas where the thin and the thick filaments are overlapping with each other, right? And then you'll also have areas where the thin filament is just kind of by itself. The green coilage or elastic filament. Now, this is what's going to allow us, when we stick it under the microscope, to see a difference in a light versus a dark band. So, let me show you another picture. Here you go. So here it is under the microscope, under your little electron microscope, and you can go ahead and you can see the Z discs being labeled, right? Because this is the border. Let me just draw a line to it. Here's the border. And then notice how they said from one Z disc to the next is going to be a sarcomere. And once again, the sarcomere is the contractile unit of the muscle cells. This is the smallest part that can contract. And then now let's go ahead and find our A band right here. Now remember, A, dark, right? When we look at the picture, notice how it has lots of black shading, and that's because it's indicating the fact that you have lots of dark, thick filaments overlapping with thin filaments. So there's a lot of proteins that are just stacked on top of each other. Find your I band next. I band right over here on either sides of the A band. This is going to be the light band. And the reason for that, why is it more of a whitish tint versus a blackish tint? Well, because there are less proteins over there. In the I band, it's a location where you will only find your thin filaments. So the light is able to cast a different view on it, allowing it to stain lighter. Whereas in the dark band, the A band, you have both thick and thin filaments filling up that space, creating a larger shadow. All right, now as I mentioned before, there are lots of different locational terms. You can see your M line, you can see your H bands. Please do not worry about that. Just make sure that you're able to describe what's happening in an A band, in an I band, and the fact that from one Z disc to the next, it's called a sarcomere, which is the contractile unit of the muscle. All right, so I said to you towards the end of part one, we're going to have this little table where we're going to kind of start from large and zoom on in. And let's see if we have a little bit more comfort level now with the individual pieces that I've sort of been discussing with you. Thick filament, thin filament, myosin, actin, like how's that all related to a muscle, okay? This is table 11.1. I really like it because what we're going to do is we're going to start off with a muscle, as in the contractile organ. So something like your shoulder muscle, like your deltoid, your tricep, your bicep. Pick a favorite muscle, whichever muscle you're interested in. And what we see happening here is that on the organ level, if we were to zoom in, we would see a few things. The first thing we would see is that it has a fibrous covering called epimyosin. There's the epimyosin. It's a fibrous covering, and this is going to allow you to separate one muscle from its neighboring muscles, okay? So it's going to just kind of wrap the muscle in this fibrous sheet, and it's going to allow it to be separated from the rest of the muscles that are in that general location. Now notice also how the muscle is not made out of just one solid entity. Instead, when we zoom in and take a look at this little illustration right here, we see that it's made out of all these bundles. And the bundle, so this little part right here, this little bundle is going to be called a fascicle. You can see right here, that's our next item, a fascicle. And inside of the fascicle, we're going to see that you have muscle fibers. Now remember, your muscle fibers are your muscle cells. So these are the individual cells. So the muscle cells will clump together and they'll make a fascicle. And then the fascicles will kind of clump together and they will make an overall organ, an overall muscle for you to interact with. Now, your fascicle has a connective tissue wrapping itself, and that is going to be called the perimyosin. There you go. So the epimyosin, my apologies. The epimyosin is the outermost wrapping, and then the perimyosin is the one that's going to wrap itself around a fascicle, and a fascicle is basically a bundle of muscle fibers, which are your individual muscle cells. Within the fascicle, notice also how you have a blood supply. Notice the red and the blue lines, right? And you will also have nerve stimulation, courtesy of this little yellow structure right over here. Now, if the organ went to a bundle, a fascicle, and inside the bundle we see that we have muscle fibers, individual cells, let's see what's happening within the individual cell itself. So that's the next one. Now, we talked about the fact that its plasma membrane is going to be called a sarcolemma. And also what we see happening is that within the muscle cell itself, you will have some smaller bundles. These are going to go ahead and be called myofibrils, right here. And the myofibrils will have your myofilaments. So they're made out of the myofilaments. The myofilaments, of course, are going to be your thick and your thin protein or filament that we were just discussing in the majority of part 1. There is also a protective fibrous sheet around the muscle fiber. This is going to be called the endomyosin. So notice how you had epimyosin outside of the muscle bulk itself. You had your perimyosin, which wraps around the bundles of fascicle. And then now when we're at the level of the muscle fiber, the individual muscle cell, we have our endomyosin. We also have other things here that we need to talk about. So we need to talk about things like, it's mentioned here with the Z disc, we have that. Endoplasmic reticulum, we have to talk about that. And we also have to talk about the T-tubules. So lots of things to still discuss. Alright, so here is a myofibril. And these are the ones that are stacked together, what we saw on the previous slide, to make up your muscle fiber, your individual muscle cell. And within the myofilaments, you should see some characteristics that you're already familiar with, which are your myofilaments, which are your thick and your thin filaments. So these are the ones that you have your myosin and you have your actin, and they're very organized, stacked on top of each other. so they can create the striated pattern that we're used to seeing whenever we look under a muscle cell under the microscope. All right, what else do we have? Oh, so we have T-tubules and sarcoplastic reticulum. And in part 2 of our lecture, we're going to discuss these two parts in great detail. But just to kind of give you a little heads up, the sarcoplastic reticulum, the SR, are going to be regions where you're able to store calcium. Now remember how calcium is important for the troponin to move the tropomyosin out of the way? So we're going to need to have access to calcium if we want our muscles to contract. So one of the ways we're going to get that is by having it being released from our sarcoplastic reticulum. The T-tubule is what we call an invagination. It's going to start on the outside of the muscle cell, and it's then going to go ahead and make its way into the interaction with the myofibrils. And what we see happening with the T-tubules, it's an excellent way to spread the excitement from the outside of the cell to the inside. So I'll definitely be talking more about that. Just a little heads up that you have your SR and your T-tubules within the same location. Now, the thin and the thick filaments, how they're able to interact with each other. Remember, they're going to allow you to form your sarcomere. Your sarcomere is your contractile unit of the muscle fiber, and it will go from one Z disc to the next. So you can see your little Z discs, and we can see the alternating of the thin and the thick filament. Now, just a little disclaimer, okay? So in cardiac muscle and in skeletal muscle, you have your striations, meaning that your thick and your thin filaments are very organized, right? They're alternating, just like we see on our little picture right here. And that is then what allows for the physical contact and the contraction. Smooth muscle does not have striations, but that only means that they still have thick and thin filament. They're just not organized in a very parallel pattern. Instead, with smooth muscle, we see that they're just kind of like thrown into the batch, and they have this very irregular pattern or different amounts of overlap that they'll have. And this then helps us explain that when we look at the contractile power, when you look at your cardiac muscle and your skeletal muscle, they'll contract in a very uniform pattern, meaning you're able to predict where the muscle is going to go when it's contracting. On the other hand, when you take a look at smooth muscle, because their sarcomeres, their thin and their thick filaments are not stacked in a parallel format, the muscle is able to contract in all different types of directions because it will all depend on where they can make contact and interact with each other. Of course, referring to the thin and the thick filament. So, sorry, I got a little bit off topic, but I just wanted to share that even though they don't have striations, that the smooth muscle does have thin and thick filaments, just in an unorganized pattern. So let's come on back to our picture right here. So gentle reminder, from one Z disc to the next, that's going to be called the sarcomere, and the sarcomere is the contractile unit of the muscle fiber. So this is the smallest part that needs to give a reaction before it's able to radiate outwards and get a larger response. And of course, within the sarcomere, you'll have your myofilaments. And the myofilaments just reference the fact that you're going to have your thin filament made out of actin, and you're going to have your thick filament made out of myosin. All right, I'm going to stop here for part 1. Take your time and review the material. Please make sure that you're comfortable with a thick filament, a thin filament, what a sarcomere is, what a Z disc is, because that's going to make it a little bit easier for us in part 2 when we start discussing the actual mechanics that go into making a physical contact between your thin and your thick filament. All right, as always, if you have any questions, comments, and concerns, please do not hesitate and send me an email, and we will go ahead and talk soon for part 2 of chapter 11.